Yesterday we talked about the glycolysis and Krebs cycle, and there was an animation. So there was no animation for electron transport chain, so I went and found one. So let's watch this one. I know, because, well, it's an important step. It produces the most ATP.
So to cover yesterday's stuff, yesterday ATP broke off, made ATP came back on, made some
ATP, yeah.
That's all it was.
But then we went through how they make ATP, how you break apart like glucose. Is it going to play or not? Otherwise, it would distort. You would never use the energy. In order to use the energy, it's got to break off that phosphate. Otherwise, there's no energy. Let's make sure we can get the...
Introducing
Okay.
the all-new by Yahoo, your favorite glycolysis and the tricarboxyl.
Okay, shh.
Oxidation of organic molecules results in the production of reduced coenzymes such as NADH.
So remember we made NADH and FADH too yesterday?
Transport chain, which is located in the bacterial cell membrane. A hydrogen consists of a proton.
In the bacterial cell membrane, it's talking about because there's some theories that the mitochondria is actually a bacteria that got ingested and is used in that's where it came from, but who knows. But it's in the mitochondria membrane here.
So this bacterial cell membrane is actually...
Yeah, so in our bodies, it's in the mitochondria membrane.
And an electron. The electron transport chain consists of a series of special electron carrier proteins that shuttle electrons from NADH to a terminal electron acceptor, such as oxygen.
So this is that pathway. Yesterday we talked about the metabolic pathway and how enzymes were in the order, the correct order, and there is a rate limiting enzyme, and that would be the first enzyme here, would be the rate limiting enzyme. The white part? This is like the polar heads of the phospholipid bilayer.
When NADH transfers its protons plus electrons to a membrane-embedded carrier protein, the electrons are sequentially carried along the electron transport chain, while the protons are shuttled to the outside of the membrane. Some of the electron carriers, such as coenzyme Q, accept a proton from the inside of the cell membrane as it accepts electrons. The proton is then transported through the membrane as electrons move down the chain. This increases the proton gradient across the membrane and enhances the proton motive force.
So it's gone against its concentration gradient, and there's a higher concentration now outside the cell, and that's what the cell is going to, or the mitochondria, outside the mitochondria, I should say. Inside, yes.
During aerobic respiration, the last carrier protein transfers a pair of electrons to oxygen at the end of the electron transport chain, and water is formed.
Well, if it's just O, the oxygen takes 2 electrons, becomes O2 minus, and then accepts the 2 pluses to make water. It's O2. See, did you notice how it was? Right. Oh, gotta go back just a little bit more.
And water is formed of electrons to...
So this is O2. There's 2 oxygens bound, and then it's going to accept 2 electrons, become O2 minus, and then it will.
So all the little electrons get kept in the cell, and all the hydrogens, or most of the hydrogens go out.
Oxygen
Yes.
at the end of the electron transport chain,
Where do those
and
little
water is
H's
formed.
coming from?
Inside the cell.
The enzyme ATP synthase utilizes the energy of the proton motive force to synthesize ATP. This enzyme allows protons to pass back into the cell and couples the energy
Phosphate.
released in the cell.
So it's taking ADP, adding the phosphate, using the energy from the hydrogen coming down its concentration gradient.
But it's not really using the hydrogen.
Right.
Just
The hydrogen gets recycled inside the mitochondria.
Process to phosphorylation of ADP to form ATP.
They use that big word phosphorylation. It just means adding the phosphorus back onto the ATP.
I have a question. Why is science have to make everything so
Oh, that must be the end. That's the end.
sophisticated? You know what I mean? Like, why can't they just keep it dumb?
That one looks like it actually names all the proteins and everything. A little more. This one was enough.
Sugar goes to ATP. Wait, there's one that looks like a video game.
That's good. You can look it up.
Right.
DNA. Deoxyribonucleic acid, and this is the genetic code needed for the synthesis of each protein required by the cell. So DNA is what codes for these proteins, but proteins are the ones that actually make your body work or make the different compounds or will do the processes, but DNA will code for the proteins. So a gene is a portion of DNA molecule that contains the genetic information for making a single protein. And a complete set is called the genome. So all the DNA in your body is called, in the cell is the genome, but there's individual genes that code for a single protein. The nucleotides of DNA form a sugar phosphate backbone with bases extending into the interior of the DNA molecule. The nucleotides of one DNA strand are compatible to those in the other strands. So we need to know these pairs, where adenine pairs with thymine, cysteine and pairs with guanine. At CG? Okay. And so exhibit complementary base pairing. So if one strand has the adenine, it's paired with the other strand thymine. And that is how RNA gets, or the DNA gets replicated and how they form RNA. What? I've never seen it. What is it?
It's about like a sophisticated society. You get to design it.
Hmm.
And it's about a guy who wasn't designed, he was actually made, and like about how he like overcame his brother, and he has like a really bad heart condition, but he really wants to go into space. And we watched that.
Yeah, that's why I want to watch it.
The DNA molecule twists then and makes that double helix. So there's the sugar backbone is the blue strand that goes along, and then the base pairs on the inside. Yeah.
Well, some of us are.
DNA replication. So each cell must have an exact replica of the parent cell during mitosis. Actually, the DNA replication occurs during interphase before mitosis begins, and then mitosis splits it. And the DNA molecule splits. Nucleotides form complementary pairs with the original strands. And then each new DNA molecule consists of a parental strand and one newly synthesized. And that's where mutations could possibly occur if there's the wrong base pairing or some crossing over and stuff. So there's where it has the parent strand is the dark blue, and then the new strand is the lighter blue. Mm-hmm. What? They should figure out how to fix mutations.
To replicate, the DNA molecule must first be unzipped and unwound. This is accomplished by the enzyme helicase. The point where the separation occurs is called the replication fork. A major replication enzyme, DNA polymerase, can only add bases to a pre-existing starting strand. A primer must therefore be synthesized before DNA polymerase can begin to make a new strand of DNA. Another limitation of DNA
Which one?
replication,
Which purple thing? This one?
No,
These?
no,
This part?
yeah, no, okay, maybe it's not purple, it's red.
Red. Oh, that was the primer codon. So that's what. No, that's what's going to start the replication. You need this codon attached, and then it will make the polymerase will come and attach. These things? That's the DNA polymerase. This one helicase, and that's just unzipping it.
one strand of the DNA, called the leading strand, is replicated continuously, whereas the other strand, called the lagging strand, is synthesized in small segments. DNA polymerase builds the segment backwards away from the helicase until the strand encounters the previous segment. A different enzyme, DNA ligase, then joins the short segments together, making up the lagging strand. Once the process of replication is complete, there are two identical molecules of DNA, and each consists of one old strand of the original DNA and one newly synthesized strand.
Okay. Why do you need to know this? This is how the cells replicate. We're talking about cells. We're learning about cells. And then the DNA is what codes for a protein, which we're going to have RNA. After this is going to code off the DNA, and then that forms the protein.
DNA replication begins when helicase unwinds a segment of the DNA and breaks the hydrogen bonds between the two complementary strands of DNA. DNA polymerase can only add new nucleotides to a free 3' end of a growing chain. Synthesis of one strand of the DNA, called the leading strand, proceeds continuously in the 5' to 3' direction. Synthesis of the complementary strand, called the lagging strand, is more complex. DNA polymerase can add new deoxyribonucleotides only to a free 3' OH. To provide a free 3' OH starting point on the lagging strand, RNA primase attaches to the DNA and synthesizes a short RNA primer. DNA polymerase 3 then adds deoxyribonucleotides to the 3' end of the RNA primer. DNA polymerase 1 replaces DNA polymerase 3, removes the RNA, and replaces it with DNA. Finally, the enzyme DNA ligase forms a phosphodiester bond between the 3' OH of the growing strand and the 5' phosphate in front of it. During DNA replication, the leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously.
Wow, that's a lot more complex, and you can see why there might be a mistake. Right, Allie? But they should fix it. They should fix their own mistakes. Okay. Let's see what this one has.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase 3 is the major enzyme involved in DNA replication. DNA polymerase 3 can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides, complementary to the parent DNA. DNA polymerase 3 can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand
They really pound this leading and lagging strand in, huh?
synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Okay, so each of those had similar information, but a little bit different extra parts in each of them. So there's RNA involved in DNA replication, and then the majority of RNA is really involved in protein synthesis, where it codes for a new protein and it leaves the nucleus where ribosomes are going to attach amino acids to it. So there's two parts, transcription and translation. Transcription is where RNA molecules are attached and replicating the DNA, and they are single-stranded, contain ribose instead of deoxyribose, and uracil other than thiamine. So instead of the AT part, it would be AU, and then CG. Messenger RNA is what is synthesized on the DNA. We shorten that to mRNA. And that is the transcription. So transcribing DNA, because when you transcribe something, you guys know what that means? You copy someone else's stuff. So you would copy, like, books used to be transcribed way back before they had the printing press. So transcription is the copying of DNA onto mRNA, a form of RNA called mRNA. Then that mRNA leaves the nucleus. Oh, here, I think this one, well, is it? This would be the RNA strand that's just being, so it unzips a little bit, just a little part, and then it would go back together after it's been replicated there. And then that RNA, mRNA, goes outside into the cytosol, and it's translated into a protein. So each amino acid corresponds to a triplet of DNA nucleotides. A triplet of nucleotides is called a codon. So like maybe ACG, that would be a codon. And that would code for a specific amino acid. So mRNA moves out of the nucleus, associates with the ribosomes, where the protein will be constructed with the ribosome. How many subunits are there on a ribosome? Remember? 2 subunits. It comes and attaches it onto the mRNA. Okay. Once the ribosome is attached to the mRNA, a second kind of RNA called transfer RNA or tRNA, it has a triplet of nucleotides called the anticodon, and it is going to pair. It's complementary to the mRNA, so it's going to pair with those 3, the codon on the mRNA, anticodons on the tRNA. And the ribosome holds the messenger RNA in position while the tRNA carries out the amino acid. So it will match up those 3, the codon with the anticodon, move to the next codon, a new tRNA is going to attach, and then we'll move on down the strand of mRNA. Wow, there's a lot of spaces there. The ribosome contains enzymes needed to join the amino acids together. So the tRNA is bringing the amino acid to the ribosome, and there's a protein that will join those amino acids, make a peptide bond. And once they're joined, that's when you have the new protein molecule. So here it's showing this is the transcription. This is the RNA, the mRNA that's being formed from the DNA. It leaves through one of those pores in the nuclear envelope. The ribosome attaches to it. These would be the tRNA molecules. So a tRNA molecule is coming in and attaching. These are the anticodon. The codon here, they're attaching. And then the amino acid that it's bringing in will get attached to this amino acid. And then the free tRNA is going to leave, go get another amino acid. And so each codon codes for a new amino acid, and the order of amino acid is what makes up that protein. Gives it its specific shape, structure, and function. This is just showing how the tRNA attaches here and makes that new bond. This is translation. Maybe. Here we go.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site, as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
It goes from A to P to E. The tRNA does. APE, yeah. So it attaches at the A site. It moves over to the P site where it will bond with the previous amino acid, and then it moves to the E site where it gets released. Let's see what this one has to say.
Most of the control of gene expression in eukaryotes is achieved by regulating the frequency of transcription initiation. Various regulatory proteins bind to DNA and affect the ability of RNA polymerase to bind to the promoter site and carry out transcription. Efficient binding of the RNA polymerase increases the rate of transcription initiation. Gene expression can also be controlled by altering the splicing of exons in eukaryotes. For example, The faster messenger RNA is processed, the greater the amount of gene product. Gene expression can also be regulated by controlling access to or efficiency of transport channels. The passage of an mRNA transcript across the nuclear membrane is an active process requiring that the transcript be recognized by receptors lining the interior of the pores. Many enzymes degrade messenger RNA. Gene expression can be regulated by modulating the degree to which the messenger RNA is degraded. Many different proteins and amino acids take part in the translation process. The rate of gene expression can be regulated by speeding up or slowing down protein synthesis. The availability of any of the proteins or amino acids can alter the rate of the protein synthesis. Phosphorylation or other chemical modifications can alter the activity of a protein after it is produced. This is referred to as post-translational modification.
Okay. And yep, I thought so. We're done.